Hi guys, and welcome to Laboratory 7 with Mr. Hopper and Ms. Rose. So we are going to work you through the concepts today of photosynthesis and fermentation. And to begin this, we're just going to walk through the opening parts of your lab manual and get across what information we can to help you with this lab. And really the first part that's worthy of consideration here is this formula for photosynthesis, and that is that photosynthesis, the formula for this is carbon dioxide plus water, utilizing solar energy, can produce glucose, or generally a sugar, not necessarily glucose, but a sugar for our purposes, and plants release oxygen. Now, you know this. You know that plants take in carbon dioxide, and using photosynthetic activity, they release oxygen, and then you take in oxygen and release carbon dioxide. So these are photosynthesis versus what we do, which is cellular respiration, are sort of opposite ends of the coin. Okay.
So we're going to look at both of those
similar
processes in this lab today.
All right. So in particular, photosynthesis and fermentation. Now, I will go through with you and walk you through these models, but these images that you can see here in your lab manual are incredibly important. The parts that you need to focus on are the thylakoids, which are the little discs that you can see here. Their membranes are what literally house the photosynthetic pigments that you'll be looking at. They are in stacks called grana for reasons that we will talk about in lecture, and this would all be in a fluid-filled chloroplast, and the fluid inside of this would be referred to as the stroma. Now, the chloroplast would be inside of a leaf, in particular inside the mesophyll of that leaf, and inside of there, they must have the capacity to get at, if I can flip my page here, this carbon dioxide, and then to vent that oxygen to prevent what's called photorespiration. So what they will do is they will have what are referred to as guard cells, and these guard cells, as you can see here and one down here, are going to be utilized to regulate the flow of gases in and out of a given plant. And we're going to look at that today on a literal leaf. We're going to see it, and we're actually going to play some games with it to see if we can osmotically cause these stomates to open and close, which is a pretty fascinating process. You see, what plants do is called transpiration. What they will do is they will pull water in from their roots. That water will come up through the plant, be worked with in the leaf, and as the plants open their stomates, the stomates will allow some of that water to leave as vapor. It's just part of how the system works in the grand scheme. You know that you can go out and break a leaf or pull a limb off a tree or something, and you can see a fluid accumulate. The plant has to store fluid internally, much like we store fluid internally using our skin. So when you breathe, you will release water vapor, and I'll point this out later, but if you hold a piece of glass or something up in front of your face, you can see the water vapor form, and that's not unlike what happens with these stomates. So plants, in very earlier phrase, when there's plenty of water around, the guard cells, which you can see there, guard cells on a stomata, or a stomate, a singular stomate, multiple stomata, the guard cells will be open when the plant has plenty of water so that they can exchange gases, and if the plant were to begin to dry, the stomates would close to prevent further water loss. So this concept is incredibly important.
And I want to mention to you that these are good lab test questions to ask you about these structures off the models that we have. These are pictures of our models. So you notice that in the lab book, it gives the parts that you'll need to know. So we didn't do xylem and phloem on the plant, but we've got basically these structures related to the chloroplasts and the pigments and the guard cells and that kind of thing.
Speaking of pigments, so we can actually extract these pigments for analysis as part of the lab today. What we have is chlorophyll A, chlorophyll B, xanthophyll, and carotene. Chlorophyll A being bluish green and is considered the more important of the two for photosynthetic activity. So chlorophyll A is numero uno, the more important plant pigment. We have chlorophyll B, which is an accessory pigment, which gives off a very harsh, or more of a mild green coloration, actually. Yeah. Xanthophylls, which give off sort of a yellowish coloration, and then carotenes, which think carrots. Carrots give off an orange to yellowish color. Now, we can extract these through a process referred to as paper chromatography.
And the word chromatography means to separate. And then the word paper tells you the medium we're going to use to separate the molecules, or in this case, the pigment molecules. So we have special chromatography paper. It's not filter paper that we'll be using. We're going to rub spinach, the markings from the spinach leaf onto it, and then use a solvent to separate the pigments into the 4 pigments that Mr. Hopper just mentioned.
So in essence, by grinding the leaf into the paper, we are depositing pigments in all pigments into that paper, and then the physical pigments have a varying attractiveness to the paper and allows us to separate them out using our chromatography apparatus. So used for plant pigment separation. And that's the end of introduction. So what we will be doing next is an observation of plant leaf and chloroplast structures using models and microscopy. All right, let's get to it. All right, what you see here, this is a chloroplast. What you need to know about the chloroplast is that this is an organelle that is used for photosynthetic activity, so this will be found inside of a leaf. Now here we have a cross-section, if you will, of a leaf, and in this, what we'll have is this spongy mesophyll, which is an area of, in essence, gas exchange. You can see stomates here and there. On top of this, so this would be a stomate with 2 guard cells. The goal would be, again, that when the plant has optimal water content, that these cells fill with water, and when they are full, they are open. And when the plant begins to dry, if it begins to dry too much, the guard cells will lose fluid and then they will close. And by closing, that prevents what we call transpiration, or the plant releasing water, okay, through its stomates. When the plant exchanges gases, it's not unlike when you exchange gases with the atmosphere. When you breathe, if you hold your face up to a piece of glass or a mirror or something along those lines, you can see the fog. This is not unlike how plants function in the exchange of gases. Now, we can see the mesophyll here yet again. So we have the spongy mesophyll down here at the bottom, more or less for gas exchange, and then the palisade up top. And in this, you can see where we were trying to label, I suppose, to be chloroplasts like this here. Now, on our chloroplast, again, this would contain these thylakoids, these individual discs referred to as being thylakoids, and in the membranes of these is where you find your chlorophyll and all that fun stuff that leads to photosynthetic activity. The stacks of these are referred to as being grana, and the fluid that would bathe all of this is referred to as the stroma. Want to add anything?
So the stroma on this model is what the yellow is. Okay, the yellow refers to the fluid, and you see it will cover around.
It would fill the whole chloroplast.
That's right.
What you see here is a stomate right there, okay? These are all actually stomates. So these are the, think of these as openings that allow for gas exchange with the plant. Now, what I'm trying to show you here is that this one is nice and open. You can see the hole in the middle. And what I'm going to try to do is I'm going to try to expose this to a saltwater solution, and that should, in the grand scheme of things, begin to dry it out. Now, when the plant begins to dry, it's a natural process, as the plant begins to dry, the stomates will close in an effort to conserve water. Now, I'm not exactly going to dry it, I'm just going to play with its osmotic balance, and that may or may not function in closing the stomate. Let's see what happens. So I'm adding incredibly salty water at the moment. We're going to see if that stomate closes. All right, I believe we have succeeded. So if you look at this now, it does not appear as open as it was before. The opening itself is much smaller than what you had seen previously. All stomata. These are all gas exchange openings for the plant. Now, the two sort of the bumps, if you will, on the side of this, so one on either side, one there, those are guard cells with the actual stomate opening there in the middle. The whole thing is a stomata, guard cell, guard cell. What we are doing here is activity 2. This is plant pigment separation. Now, from my perspective, what we will be doing here is part of a larger experiment. Okay, the goal of this experiment is to take a given leaf, like a spinach leaf of this fashion here, and to extract from this the pigments, as shown in your figure here, that give rise to photosynthetic activity. Now, there are numerous of these pigments, not least amongst these carotene, xanthophylls, chlorophylls A and B. Okay, there's more in the world than this, but this is what we're dealing with. And what we're going to try to do is extract these, and you would realize that the colors that they demonstrate when run through this chromatography apparatus we'll be explaining here in just a minute, that would represent the color of light that they reflect, and that they would absorb light from other wavelengths, not the color that they reflect, if that makes any sense whatsoever. So what we have here is our chromatography apparatus. Ms. Rose, would you explain what we've got?
Okay. This is the apparatus ready to use. We're going to put solvent in it and then get the experiment running. You can see the green is where I've rubbed the spinach leaf onto the paper, but I wanted to show you, it's a little bit tedious getting it set up, so I wanted to show you what I've done prior to that. And I've got my paper. I drew a line where I'm going to rub my leaf onto it. I've had to trim my paper so it does not touch the sides of the glass. You've got to make sure that the solvent runs through the paper, through the pigment, through the paper, up, the test tube, and not all of a sudden touching the glass and solvent running along the glass.
It plays games with it on capillary action.
Yeah. So anyway, here's a spinach leaf. I used a penny to rub this and did it multiple times. And so this is the setup.
So what we get is this dark green line. You can see the line where we started there. This dark green line across the paper, this special chromatography paper in the grand scheme. And what's going to happen is when you expose this to our chromatography fluid, that fluid will run through the paper and pull the pigments with it, depending upon their connectivity to the paper and their solubility in the solvent.
And I want to mention to you that this chromatography solvent is very flammable. It's an ether acetone mix, and so we have to be very careful. We have to get the ventilation going in the lab. It's not something that you would do at home in your kitchen. Used solvent has to be disposed of properly, so there are a lot of precautions that we have to take in this particular lab.
So you'll probably hear the fans running while this is taking place. All right, and we will pick it up the next time through on the preparation and running of the solvent. What we have here is our plant pigment separation. This is a paper chromatography apparatus as we had set up previously. You can see where we began the process down here, and we've had our chromatography solution of ether and acetone soaking through the paper, and as this has happened, it has carried these pigments with it via capillary action. Now what we are supposed to see here are 4 pigments: carotene, xanthophyll, chlorophylls A and chlorophyll B. Now, would you like to walk us through this?
Okay. All right. So going from the bottom, we've got chlorophyll B. Now you can see we have a little bit of a peak here. It's not just a band that's totally horizontal, but the chlorophyll B is right there. And then you've got chlorophyll A that comes up here. And then we've got xanthophyll, the yellow. And then usually there's a little bit of a gap. And then the carotene comes off after that, so we think the carotene is right about here. And usually you see a flash of a muted orange. It's not bright orange for carotene. Okay, so there's the 4 pigments: chlorophyll B, chlorophyll A, xanthophyll, and then carotene right here. And then the solvent front, which is something that we have to measure, is right here at the top. And it will evaporate quick, so we're paying attention to that so that we could measure that.
Ruler against it?
Yeah, you gotta.
Like so?
Yeah, so the solvent front has moved 126 millimeters, 12.6 centimeters. I think in the lab book it tells you to measure millimeters. So 12.6 would be 126 millimeters.
So good. All right.
So with the table that's here, what we want you all to pay attention to is the order in which the pigments come off, okay, from the where the spinach line was. So again, chlorophyll B, and what you're learning from that is you've got a question at the top of the next page that says which pigment has a greater attraction for the paper. So what we're really asking you is which pigment likes the paper the most. And if it likes the paper, is it going to move the furthest or the least? Okay, now if you like the paper, then it's going to move the least. Okay, and another question that we could ask that's not here but could be asked is which pigment has a greater attraction for the solvent? Okay, so if the pigment likes the solvent, do you think it's going to not move very far, or it's going to flow with the solvent up the chromatography paper? And that would be the answer. So carotene likes the solvent the best, while chlorophyll B likes the paper the best.
Hi guys, in my infinite wisdom I managed to not actually record an introduction for this portion of the lab. What we're going to be doing is activity 3 using a respirometer to measure fermentative reactions. Now before I really go into the details here, there are two forms of respiration that are worthy of our consideration. Now up to this point in the lab we've been talking about photosynthesis. Now we're moving on to respiratory processes. This would be completed in mitochondria, for example, if we were doing what's called aerobic cellular respiration. Aerobic cellular respiration is the best way to make as much ATP as is physically possible from a given glucose molecule. But what if a cell or an organism doesn't have oxygen available? Well, there is another path that can be used, and that path is a fermentative pathway. Now, fermentation is a good way to make some ATP in emergency situations, and some organisms like the yeast we're going to be using today really specialize in that, but it doesn't make nearly as much ATP as does good old-fashioned standard aerobic cellular respiration. So what you can see here on the page I've provided for you is the formula for how yeast break down glucose and make ATP. You can see that the glucose is broken down into carbon dioxide, alcohol, and just a very small amount of ATP molecules. Alright, just a very tiny amount compared to in the 30s for aerobic cellular respiration. But in this process, we can use the knowledge of what we have here to figure out what the yeast will do based off of different sugar molecules that we will be providing for them. So what we're going to do is we're going to use a special lab apparatus called a respirometer, and this respirometer gives us the ability to measure the amount of carbon dioxide produced by yeast, which is the organism we're using in this situation, and the amount of carbon dioxide produced is a tell of how effectively they break down certain different sugars. And the sugars will be shown as we progress through this. But let me just make sure that we're clear. This is a process used by organisms in aerobic situations where there's no oxygen present to make a little bit of ATP, more or less in like an emergency situation for us. Okay, so not the most optimal way of doing business, but a way of doing business all the same, and still a respiratory pathway.
What we have here, folks, is a little flask that has fermenting yeast inside of there. You can tell that it's fermenting because you can see the bubbles down the base of this. And what I'm going to do is I'm going to strike this match here, and we're going to stick it down in there and just see what happens. You know, we may not get anything taking place, or we may see an effect, and I hope that we do. Okay, so here we have our match. It's burning, and I'm going to take that and I'm just going to stick it right down inside. Whoa, look at that. Woo. Now, why did it go out is the question. Now, previously, we did a Catalase experiment, and that Catalase experiment was releasing oxygen, which made the match flame up. By comparison, this is a fermentative reaction, and fermentative reactions release carbon dioxide, and carbon dioxide just kills that match immediately. It prevents any oxidation to the wood, and thus the match goes out. That clearly demonstrates that we have a fermentation reaction taking place, the production of carbon dioxide. The making of respirometers. This is the tool that we're going to use to demonstrate fermentative reactions. Now what Ms. Rose has done is she's taken her smaller test tube here and has filled it. I don't know if you can see, but it
I'm
is
gonna add another
it's
drop.
very, very, very full. So again, water is both cohesive and adhesive, so it's stuck to the glass internally, but the water molecules are cohesive to one another, so they are literally attracting one another with hydrogen bonds and holding together to form that bubble you see at the top, not unlike the way that water beads up on the hood of a car that is well waxed. So now to complete this process, Ms. Rose is going to place this smaller test tube into the larger test tube, and once she has pressed that to the surface, she's going to invert it in an attempt to keep the amount of gas present inside the small test tube as small as is physically possible.
And you see, this is really a magic act more than anything, because you don't want a very big bubble, and if you make too big of a bubble, you got to start over. So we have little tricks.
Just a little marble.
Yeah,
just
just
a
a
little
little bit.
marble at the top. And so this is a test tube, inverted, completely full of fluid with a very small bubble inside of there. Now, if this was a sugar solution with yeast internally, and it was incubated at a temperature, you'd expect that yeast to then ferment the sugars present in the fluid, and that fermentative action, as we just showed, releases carbon dioxide. And that carbon dioxide would make the bubble inside of this, since it's trapped, grow larger and larger and larger.
So what's important to understand is when you created this bubble from making this respirometer, it's a bubble of air, okay? We want to measure the CO2. So we have to subtract the size of this bubble from what we find the result in the experiment, the size of the bubble in the experiment. So that's why we're being really careful here to keep this bubble small. And we're going to we did this for practice, and then we have the respirometers we're going to make. I've put sugar 2/3 of the way. You can see in your procedure on page 111 in your lab book.
So you can see what the sugars are here.
Up at the top, it tells you to mark that 2/3 and then fill to that mark with the sugar. So we've got glucose in the first one, maltose in the second, sucrose, and then water, distilled water, our control.
I was about to say, Ms. Rose, why would we use distilled water for the last one?
Absolutely. It's a
Well, that
control.
is a scientific control. We're going to use that to demonstrate any variation in bubble growth just as a result of the water being warm and potentially having a little gas formation.
So now what we need to do is you can see there's about 1/3 of the test tube left. We're going to fill that with yeast, our yeast you saw just a minute ago. And we're going to overfill it. We're going to invert that. And we're going to have our respirometers ready to go. We will have measured the size of the bubble that we create, the air bubble, and then we're going to put them in the water bath, the warm water bath, and let our reaction happen.
All right. So what we have done is we have gone through and added our yeast solution to all of our test tubes here, the small ones. We're going to invert those and place them into their accompanying larger test tubes, and this forms up the apparatus referred to as being a respirometer. Okay, we're going to be able to measure fermentation by using this lab tool, a respirometer. Now, this is done all the time in microbiological laboratories to demonstrate the capacity of certain bacteria to ferment certain sugars. Basically, are they a maltose fermenter or are they not a maltose fermenter? These are ways that we can identify a bacterium's type.
So we're going to measure this bubble in terms of millimeters. And you can see I'm having to kind of look through the larger test tube to see
Can you hold it vertically?
the bubble in the smaller test tube. So it looks like it's about
3 millimeters.
Three. I'd call it 3.
That is
And
the
this is
2%
a water.
sucrose.
Yeah.
Oh, that's a water?
I did the
okay,
water
that's
first.
the water. Okay.
Okay. All right.
All
So
right.
we're going to finish the others, and we'll have our measurements of the air bubble to put in the table.
Can we just put it in there for a second? Just for demonstration purposes. Bear with me. All right, we have just pulled these out of the incubator after 15 or so minutes, and we're quite surprised to find that they are just blown up, man. Look at the amount of gas production that's taking place here. Like we've tried to use pipettes, as you can see here, to pull a lot of the fluid off, but they were just completely blown out as it was. So what we have is glucose here, all right, maltose, which is here, and then sucrose, which is there. And overwhelmingly, what you can see is the glucose, I mean, it's it's almost entirely emptied. And we're going to try and see if we can get bubbles out of this in a second. Second, here is maltose. And Ms. Rose, can you point? What you can see there is that it is not completely blown out. The maltose certainly has not worked remotely as well as have the other two. And here is sucrose. You can see that the sucrose is virtually blown out as well. So what we're going to do is we are going to measure these by hand and then come back with some data for you that we can use for some numerical calculations. So here is our data from the take home here. So this is going to be glucose, maltose, sucrose, and water. And Ms. Rose, would you like to explain to us what you see here?
Okay, so what we did is we were able to measure the final height, getting down into all the way down through that respirometer.
We had to do
that
some
small
work.
tube.
We had to do some
Yeah.
work to figure it out, but we figured it out.
We feel like we could see the bottom of the bubbles. And so we measured that, and then we had to subtract from that large bubble, because that large bubble was from fermentation, right? So then carbon dioxide would be what we were detecting. That big bubble was due to the production of carbon dioxide during fermentation. So we had to subtract away the air bubble, okay, to give us the net change. So this would be CO2 only. This would be CO2 plus
In
air
essence, what we are measuring here is carbon dioxide production. And carbon dioxide production is going to be used as a surrogate here for the capacity of our yeast to ferment the sugars that are present.
That's right.
So what we find is that glucose probably is easiest for our particular yeast to ferment. Maltose is much more complicated for our yeast to ferment, and sucrose is somewhere in between the two.
Yeah.
And again, there's a little bit of a net change here in the water, as you can see. Not much of one, but a net change. That could be potentially due to just an artifact of the way in which we were doing our business, or perhaps the suspension of yeast had a little gas in it when it was placed in and it settled out. But one thing or another, this sort of gives credence to these values and shows that we have accomplished something.
And then keep in mind, from the fermentation equation that you saw at the beginning, is that yeast do work on glucose. I mean, that is their primary substrate. So you would expect to have the glucose to be the best reaction there.
All right.